Hello everyone, welcome back to the channel. Enzyme replacement is a medical treatment used for certain inherited enzyme deficiencies, typically in conditions such as lysosomal storage disorders. The therapy involves administration of a synthetic or recombinant version of the missing or deficient enzyme to restore its function in the body. The process begins with generating DNA fragments, which might include the target gene we want to study or manipulate. These fragments were created using specific enzymes called restriction enzymes. Next, we isolate plasmid DNA from bacterial cells. A plasmid is a small circular DNA molecule that is separate from the bacterial chromosome and can replicate independently. From the generated DNA fragments, we select the desired gene of interest that we want to insert into the plasmid. The plasmid DNA is cut open using the same restriction enzyme. This ensures that the ends of the plasmid and the DNA fragments are compatible for binding. The selected DNA fragment is inserted into the plasmid at the cut site, forming recombinant DNA. The enzyme ligase is used to seal the bond between the DNA fragment and the plasmid, creating a stable recombinant molecule. Then, the recombinant plasmid is introduced into host cells, often bacteria. These cells will take up the plasmid and begin replicating, producing multiple copies of the recombinant DNA. Inside the bacterial host, the recombinant DNA plasmid begins to replicate. Since plasmids are capable of independent replication, multiple copies of the recombinant DNA are produced. As the host cell divides, the recombinant DNA is passed on to its daughter cells. This ensures that each new bacterial cell carries the recombinant DNA. Finally, through repeated cell divisions, a large number of bacterial cells are produced. These cells form visible colonies, all containing identical copies of the recombinant DNA molecule. Once the correct bacterial colonies are identified, the recombinant bacteria are cultured in large quantities. The host cells can express the desired gene from the recombinant plasmid, producing corresponding protein or product or enzyme. So, here's the thing. Enzyme replacement therapy is still a work in progress. It's cutting edge, but it's not perfect. It can be crazy expensive, and there are still challenges to overcome, like making it safer, more accessible, and more efficient. But think about it, this is the kind of science that is shaping the future. We're talking about curing genetic diseases, producing life-saving medicines. And with this, we come to an end of our today's video. If this video intrigued you, don't forget to like, subscribe, and join me next time as we uncover more mysteries of medicine. See you next time.